Welcome to this workshop. As you just said, we will be recording this because we know there are a number of people who wanted to be here that couldn't make it. So we'll forward the recording to them. We won't circulate it any more widely than that. So just to people who have actually signed up for this workshop, and those of us who are here, of course. So this workshop is about problem solution validation for synthetic biology startups. And what we're going to do today is we have a guest speaker whom I'll introduce in a moment. We'll have some questions and answers with the guest speaker, and then we'll actually play with a few tools that might help us do this problem solution validation. So the goals of this workshop, I guess, are twofold. Firstly is to give you some useful information. Secondly, maybe there are three goals. Secondly, to give you some information about the SynBio10x Accelerator program. And thirdly, to give you a little bit of a flavour for what that program is actually going to be like. The exercises that we'll go through today will be somewhat similar to the ones that you would go through actually in the accelerator itself. Before we start today, I'd just like to start as we always do with these workshops. I'm based in Western Victoria, so I'm connecting in from the lands of the Wadawurrung people. They've been the traditional owners and custodians of these lands for many tens of thousands of years. So I'd like to acknowledge that. I'd also like to pay my respect to their elders, past, present, and future, and elders of other Indigenous communities who might be with us today. So here's what we're going to cover today. Firstly, we just want to talk about why this topic is important. In particular, we want to talk a little bit about the SynBio10x program, what it offers to SynBio founders or people who think they might want to be SynBio founders, and how in particular this problem solution validation fits into those workshops. Then we'll get into the meat of the session. We'll have a discussion with our guest speaker, Peter French, whom I'll introduce in a moment. And we've invited him to talk about the challenges and opportunities for SynBio startups in wrestling with this problem of custom solution validation. We want to play with some lean innovation tools to help you conduct this custom solution validation, and talk about how the SynBio10x Accelerator program can help you do that. So just want to check that aligns with what people were expecting for this evening. Anyone got any comments or questions, any additions, suggestions, other things you'd like us to cover this evening that are related to this topic? All right, if everyone's pretty comfortable with that, I might hand over now to Jason Whitfield, who's going to tell us a little bit about the SynBio10x program.
Thanks, Jonathan. Yeah, hi everyone. Kia ora. I'm coming to you from Bidjigal land. So I'll just share my screen here. Now we're into the third run through. For those of you who have come to all three sessions, you're now getting to watch me do this for the third time. So maybe I'm a little bit faster than usual, but probably not. So I'm Jason Whitfield. I'm the program manager for SynBio10x, which is UNSW Founders' new accelerator focused on helping early stage synthetic biology and biotech startups. Firstly, what is Founders? It's Australia's leading university-based entrepreneurial support program. Thanks for asking. At Founders, we help entrepreneurs translate their ideas and research into startups to help them deliver commercial outcomes. And besides being the best, what sets us apart is we have a really comprehensive support program, especially for staff, student, alumni of UNSW, that helps everyone right from curiosity through to scale up. And this scale up includes the 10x accelerator programs. And really, I like to think of us kind of like this little GIF on the right here, this sun, that we are kind of a slingshot where people come to us, the rocket, and we basically help them gain momentum and send them on their way and support them in their journey as founders. So one of the questions we often get asked is, why synthetic biology? Why is this the new accelerator you've created? And SynBio, or kind of biotech more broadly, is a space that has huge commercial potential. Last year, there was around 18 billion in SynBio global investment. And this is something that places like CSIRO and ACOLA have both identified in a number of roadmaps as being high priorities for Australia, where we have a lot of kind of great talent through our research in universities and CSIRO that kind of all align on these issues and can have great impact, but they often don't have places that they can go to. And one of the other reasons is we want to create an accelerator for this is we recognize that in this sector, you can't just work on this in your parents' garage legally or kind of just infrastructure wise. You need a lot of specialist infrastructure in terms of equipment, chemical reagents, so forth, to actually get in the lab and do that product development and scale up. And we recognize that UNSW Founders, being a university, actually is well positioned to address that. And that really leads me into why 10x. So UNSW Founders has a really great track record of creating really good accelerator programs, so our flagship industry agnostic 10x program, and also through the national program Health 10x, which I think is now into its 4th cohort. And in now teaming up with the School of Biotechnology and Biomolecular Sciences, or BABS, and the RNA Institute, we're able to kind of create a unique accelerator offering that takes that proven business coaching, global mentor network, and strategy advice and combine it with the provision of 6 months access to PC2 lab space and the access to R&D facilities and expertise that's at UNSW. The other big thing about this accelerator is that CSIRO's VC, Main Sequence Ventures, has kind of given us a big vote of confidence and a thumbs up, and they're partnering with us to provide $120,000 initial seed investment to each of these 6 startups that we accept into our accelerator. And I think that's a real vote of confidence, not only for our program, but for the potential in synthetic biology sector in Australia, and kind of how they see the role of the bioeconomy in Australia's broader economy in the future. So just quickly, like, why this session? I'm not really going to give too much away here, but I think we really wanted to give these workshops were created largely to give a kind of tiny taste of the kind of content you can expect from the 10x program and topics we might cover, and how we would kind of take a lot of business concepts and things like that and give you that kind of specific biotech spin as well that's relevant to a lot of you here. But also for this one, we wanted to build on the previous sessions, right? We started with how to raise capital, and last session we had Phil Moore come in and talk about customer discovery. So it's understanding who your customer is and kind of how you might go out there. And he kind of gave a few examples of companies in their portfolio that have done really well, like Samsara Eco for one. But now in this problem solution validation, you kind of know who your customer is and you kind of know what they want, what their problems are. But the question is actually, how do you get there? How can you create that product and like roadmap your way there experimentally in terms of business as well? And I'm sure Peter French is going to give us a pretty amazing rundown on that. So just to summarize kind of what you get from SynBio10x, there are 6 spots available for SynBio startups, and we're providing 6 months of support in terms of PC2 lab space, the UNSW R&D facilities, the proven business coaching that I mentioned, and also we don't just kind of bring you in, give you all this stuff, and then send you on your way and never speak to you again. We actually really love to stay in the loop and kind of stay with you on your journey and helping where we can. And there is a new program coming to UNSW Founders called Scale Up, and I'm happy to talk about that more at the end of this session. Through the program, you also get the one-on-one meetings with that entrepreneur in residence. And once you know it, that's Peter, who's talking in this session. So you actually get to see some insights into the kind of crazy good support you're going to get from him. And I don't really want to recite his CV too much ahead of Jonathan doing as well, because I think we'd probably have to add a whole nother session if we were to do that. So, but in terms of the other things you can get from this session, from SynBio10x, we have a global mentor network of over 500 people across almost every industry you can imagine that we can kind of link you to. And I think that's something that's quite exciting for biotech companies, is we can link you to people in the biotech space, but also we could link you to kind of digital tech companies, and you can kind of get some really interesting insights and objective insights from mentors that can help you really grow your business fast. As I mentioned, there is the $120,000 investment from Main Sequence via SAFE, but Founders also gives a $20,000 investment. So each startup that comes in, we've got $140K straight off the bat. And also there is potential to access $30,000 in service vouchers from Bioplatforms Australia to be used at any one of their nodes around the country. And if you don't know what Bioplatforms Australia is, they kind of run a series of, or manage a series of nodes of analytical services. So if you're into any of the omics and you're a startup in this program, you could say, apply to them and say, we need to use your facilities. And then they would, if they approve that, they would give you access to 30 grand worth of those services. And what's really great about this accelerator, amongst many others as well, is that you go through this 10-week program and it culminates in a demo day where you really get to show off and pitch to investors that we bring in and kind of network with them and show kind of the growth that you've gone through in the 10 weeks. And I can assure you 10 weeks doesn't sound like a lot to scientists, but I can assure you the amount of growth that you'll go through after this program is unreal. It'll be night and day between where you were when you started and your outlook when you finished the 10x accelerator. So now that you're getting all excited and you're wondering, am I the right person for this program? Should I be applying? Well, if you're in Australia or you can start an Australian registered company, yeah, you can totally apply. If you're engineering or using biology to tackle big problems in sectors such as ag tech, envirotech, healthcare, energy, bioproduction, or if you're into the new term precision fermentation, definitely you fit the bill for SynBio10x. The only other things we want to know is that you have a clear IP status, and by that we mean that you have a license to operate and exploit any intellectual property that your company would be using. Ideally, we'll be looking for someone who's kind of a proof of concept, and that can mean that what you're trying to do is established and there is a real solid fundamental grounding for what you're trying to do. You don't necessarily have to have been the ones to do it yourself, although that would help your chances. But we don't really want you to be in those really early days of exploring a concept. We really want you to understand what you're doing and have a clear path towards creating a product or service. And of course, we want you to be able to commit to the program. 10x is divided into 2 phases, a 4-week pre-accelerator and a 10-week accelerator. Now, the 4-week pre-accelerator that kicks off the end of May through to June is optional, but super highly recommended. It's where we really give you a lot of the core tools that you need to get the most out of 10x accelerator program, which starts at the end of August. So I said a couple of those key dates there, but the main one you need to know right now is the applications close on the 15th of May. So if you're thinking about it, submit an expression of interest now, and you'll get a look at the form and all the details you need to fill out, because there is quite a bit of information we're going to need from you. But if you need help or any more information, just reach out to me. My email is in the slides here, and you can easily find it if you go to our website using that cool QR code that I've put in the top right corner there. But the other dates, as I mentioned, pre-accelerator, 30th May, 24th June. Man, you can see how many meetings I've had today. My voice is going hoarse already. We'll run interviews generally around mid to late July for the program for the top 10 teams. So pre-accelerator is 15 teams. We'll interview the top 10, and then the final 6 are selected to run through the accelerator from the 18th of August to 26th of October. And that's really all there is from me. Thanks again for coming along to these sessions. I really hope you guys are getting a lot out of them. But if you have any questions, you want to join us, go to the website. My email's right there. Or if you're really convinced, you can go onto that Airtable link there and apply. And that's really all there is from me. I'll hand it over to Jonathan and Peter. Cool.
Thank you, Jason. So as we mentioned before, the meat of today's session is really to talk about this whole concept of problem solution validation, and particularly to focus on what makes that validation particularly challenging for startups in the synthetic biology space. And as Jason mentioned, we are very, very fortunate to have with us this evening someone that has a huge amount of experience doing exactly that, and that is Dr. Peter French. It would take a long time, as Jason said, to fully introduce Peter, but just a few kind of career highlights. Peter's a biotechnologist. He's got huge amounts of experience in research, basic and clinical research. and in the biotechnology industry, both in Australia and the US. He's been in this field for over 40 years, in particular in cell and molecular biology. For the first half of his career, he worked in research, in academic research, and the second half of his career has been more on the translation side of things, the intersection of scientific and commercial, to bring benefits of innovation to the broader world. He served as both an executive and non-executive director on boards of a number of public and private companies in the last 20 years or so. He has a really strong track record of successful business creation, capital raising, growth and strategic leadership of private and public companies in this kind of space. So it is with great pleasure that I'd like to hand over to Peter to talk to us about the unique challenges and opportunities for SynBio startups in doing custom solution validation.
Thank you, Jonathan and Jason. Really, it's a pleasure to be here and to share with you some of the experience that I've had over the years in taking an idea from a laboratory concept through to a business plan which will attract serious investors to develop it. Look, SynBio and biotechnology in general is a high-risk business. You know, you have an idea that's come out of some laboratory work, and you get excited about that. But to take it from there to the final market to get a person to buy it is a very long and risky business. Whether it be a drug, whether it be an organism that can change agriculture, whether it be a new diagnostic test, every single discovery that has the potential to be a commercial product has a series of challenges. And you know, you need to understand that pathway. But let me just give you a little bit of my own background. When I was a young academic, I had a couple of ideas. First one was, this was going back before most of you were born probably, but in the 1980s, wine casks were a big thing. So for PhD students, buying wine in casks was a great idea. I had the idea to put water in wine casks, and thought that could actually be a really good product. And then one day, about a year later, I found, or 2 years later, found it on the shelf in Woolworths. I thought, damn, missed that idea. Then I had an old house in Balmain in Sydney where we had rising damp. And I thought, if only I could get, I can't get cross-flow ventilation, but if only I could get some fan, but how would I power the fan? I know, solar power. I thought a solar-powered fan to get cross-flow ventilation. 2 years later, someone invented that. I thought, damn, the next idea I have, I'm going to work on. And the next idea I had came out of my work at St Vincent's Hospital in Sydney, where I was the principal scientist. And it gave rise to Cryosite, a company that today is still around and stores stem cells at ultra-cold temperatures and other materials at ultra-cold temperatures. I had the idea, I floated it with a few people, most of them said it'll never work. It was in 1998, we launched it on the ASX, and it pays dividends today. So if you have an idea and you think it might work, go with your gut, give it a go, because yeah, it's difficult, it's challenging, and it may not work. And most people will tell you it won't work, because most people that you talk to, in my experience at least, don't really think that new ideas will work. The usual approach that I've had in my life, and I've had a number of companies that I've started from scratch, is if it's a good idea, someone else would have thought about it, or that's not possible, it can't work. However, you are the expert in your particular area. If you think your idea has legs, then it's worthwhile going for. So next slide, please, Emily. So, okay, you have an idea. Who's the customer at this stage? Well, it's you. And this doesn't cost any money. You can do this for free. Is it just cool science? Ask yourself, is it just cool science? Or could it be a commercializable product? And be really tough on yourself. If you think it's a commercializable product, ask again, what's the potential market? Is there a demonstrable need in that market? And importantly, as Jason's already covered, is there already a patent that covers the idea? Now, if the potential market is big and there's a demonstrable need for it, and you've done a search, and you can search a number of databases, including the US Patent and Trademark Office database, USPTO database, and you've done that search, then it's time to take the next step. At this stage, you should have kept it to yourself. Don't publish it. Don't talk about it in seminars. Don't talk about it over a dinner party. But if you get to that stage, you think, okay, now what do I do next? Next slide. You need to think about if it's patentable, and you should talk to your technology transfer office, whether it be the university or the organization that you're working with. Now, this is where you need some data. First of all, for a patent to be successful, the idea needs to, or the concept needs to be patentable. And there are some tests around that we can talk about later. The idea needs to be novel. No one's ever thought about it before, and it can be quite specific. And non-obvious, which means that putting 2 and 2 together doesn't give you 4. And importantly, it has to be useful. There's no point inventing something that has no commercial value. So a patent has to fulfill those 4 criteria. And if it does, then you need to discuss with the technology transfer office what the patent application needs. So clearly it needs a detailed description of the idea. And for example, it might be a gene sequence and of a novel gene that, or of a gene that has a novel function. It needs to have an inventive step, and you need some evidence that it's viable. So this is where the customer, again, is you and the technology transfer office. And the data that you need is to demonstrate that your idea actually can work. Now, you don't need to demonstrate that the idea can work, in the absolute N situation. It can be laboratory in vitro data, but you need some evidence that the gene, the microbe, the engineered organism can produce an outcome, actually can produce that outcome in vitro. And you can then imply that it may also be useful industrially. So it's sort of minimal data, really. Now, it's a tragedy, really, that many investigators are not aware of the damage that can result from failing to patent. So this is a really critical step. And I know that some academic researchers who have no interest in profiting from patent licensing believe that they're doing society a service by publishing their unpatented discoveries. But seriously, the cost of taking an idea to the market through the regulatory hurdles is enormous, in the tens to hundreds of millions of dollars. And to be able to raise that sort of money, you need to be able to have exclusive ownership of the idea for a period of time, which is what a patent gives you. If you publish a result that provides that data, then anyone can do it, but no one is likely to invest in it. It's beyond governments at the early stage to do it in terms of that sort of money. So patenting is a critical stage in the commercial process. And you have to be careful about patents. Even a seminar in your department can make your patent invalid. So be very careful about discussing your idea or invention too widely before you've got a patent application filed. And to do that, you need to actually have enough data to show that it's viable. But the good news is that once you have even a little bit of data, you've then got about 18 months to add to that patent before it gets published. So you can do further research work. But the minimal data that you need for a patent is to show that it can work. Next slide. So let's just think about an example of this. Now, this is just something that I've dreamt up, but let's assume that you've got that you found a gene from a bacterium in the Dead Sea that could assist plants to grow in salt-affected land. So what in vitro data would I need to demonstrate that this is viable? Well, you could establish that the bacterium has functionality, and presumably you've already done that, that it is functional in terms of being able to allow the bacteria to survive in a high salt environment. And you've done that by perhaps by knocking out or turning off or silencing the gene and showing that in a high salt environment it doesn't grow, but the organism with the gene can grow in that high salt environment. So that's encouraging. That's sufficient data for a patent application to show that in fact this has the potential. Clearly, you haven't put it into plants. Clearly, you haven't put it into plants that are growing in high salt environment, but you've got a little bit of time, and you have some data that you can use to file that patent application. The next slide. I think you've gone back. I think you've gone. Yeah. So the next stage is, okay, you've got the idea, you've got a little bit of interesting data, you've got a patent application filed, but you need some money to go forward. So the customers in this area are the early stage investors, angel investors. And they're your customers. I mean, ultimately, the customer for the gene that I've talked about, or is an agricultural company or farmers. But at this stage, your customers are your early stage investors. You have to convince them to put some money in to help you advance your product. So what data do you need for that? Well, you need some evidence that it can work, and that evidence can be what you've used for the patent application. So that's good. You need to show that you've got control of the patent application. You need to have an idea of what the market size is. And then you need a commercial roadmap and budget for what you will do with that money. Now, the 10x Founders program can help you with that commercial roadmap. And the idea is that we can provide you with the assistance you need to be able to provide a convincing case for investment. Obviously, the more data you have in terms of it being close to being useful in the actual environment that it will be used in, the better. So if you were able to transfect a plant with the gene and show that it can grow in an environment that is salt affected where previously it couldn't, then that's clearly a very convincing case. However, to do that can require a fair bit of funding, and therefore clearly you need to have enough data to be able to convince people to take the risk, to invest in your company or your idea, to be able to test it in that sort of environment. The more data you have, the more money you can raise, and the less of the company you have to give away. The less data you have, if you raise money, it's at a higher risk, and therefore the investors will want more of the company. So it's really a sort of a dance here, where you need funding to be able to reduce the risk of the technology not working, but at the same time, you then have to be able to be prepared to give away more of the company to be able to achieve that data. So it depends on what your idea is and what your ultimate aim is in terms of how far you want to take the idea. But this fundraising issue becomes an ongoing challenge, and it's exciting. I mean, you may never consider yourself to this stage to be a salesperson, and I never did as an academic scientist. But when you have a passion about your idea and you really believe in its potential, it's a real pleasure to go and present it to potential investors. And the more questions they ask, the better it is. So raising funds is a really exciting part of this process, but you need some data to be able to answer the questions, to help the potential investors understand the risks they're taking. So let me then skip ahead forward. I was a scout for a biotech company a couple of years ago, and they asked me to find new interesting opportunities to in-license some potential technologies for them. And so I went around to a number of technology transfer offices in universities around the world. And next slide. So I looked at 50 opportunities that were out there that kind of were in the field of interest that I was looking for. So this is taking from the other side, from saying I'm the customer, I'm looking to license in a technology that has a particular area of interest that I'm interested in, to be able to invest in it, to collaborate, to help drive it forward. What am I looking for? So my first filter is that there's a patent on it that's either granted or there's a patent application. So that knocked out a small number of candidates. My next filter was, no, that's gone too far, just go back. My next filter was that an exclusive license could be obtained by me to that patent application. And again, that knocked out a few more candidates. The next slide. Is there solid preclinical data or laboratory data to show that the technology is viable? Again, that's part of the patent application data, but is there more? Next filter was, what's the market size? Does it address a significant market need? If it's a niche market, the amount of money required to take it to the market may not be a compelling profitable case. So if it addresses a significant market need, then that increases the priority. The next filter included lots of things like, was there some real world data? Are there plants that can grow in salt with that gene? The raw data needs to be available for review. Is there freedom to operate? Are there other patents that infringe? Any peer-reviewed publications? An understanding of what the regulatory pathway looks like. How near to market is it? Does the inventor have a track record of commercialization? And does the institution have a track record? So all these things meant that it narrowed down to 2 candidates, of which both were licensed in and are being worked on today. So these are things that are further down the track. But bear in mind that this is a long process where as each stage as you go, the customer changes. And as the customer changes, the data need changes. So yes, you might have a great idea, which is really cool science. And you've done your work around, there is a significant market need and there's apparently a freedom to operate in the area. What data do you need now to check or to convince us that it's viable? That's the first and early stage. So with that, I'm happy to throw it open to questions. I mean, it's a very broad question. Each of the projects will no doubt have their own specific areas and needs for data gathering to convince someone that it's viable. So I've been fairly generic in this, but happy to address to address any questions that are specific around your needs. Thanks.
Thanks very much, Peter. That's a really great starting point, I guess, for what could potentially be a really exciting discussion here. We've already got one question on the chat from Lucas, just asking about and with provisos, just asking about the size of investment or fundraising at this kind of early stage for SynBio startups. Yeah,
I think look, the answer is as much as possible that you can live with. You should have a clear idea as to what the cost of each stage will be. So if you know that the cost of getting to the next proof of concept is $140,000, then you can justify that. But everybody knows that it normally costs a little bit more than you expect. So a buffer is often a good idea because otherwise you'll run out of money before you get, you're likely to run out of money before you get to the critical point. Most investors will understand that. And most investors will say, well, look, this is clearly how much you need. But, you know, we would like to be able to add a bit more to that. And of course, the more that the investors add, the more the company that they take. So it's up to you to have a view as to how much you can live with giving away the company at which stage and how much you need to be able to raise to do the work. So it's a dance.
Thanks, Peter. And as Emily said on the chat, if you'd like to ask questions via the chat, please do that. Or if you prefer to just raise your digital hand and you'll take yourself off mute and ask the question verbally, that's great too. Not everybody is is on the bus like Lucas is. Peter's asking, in the context of Australia, how do you see the role of venture capital compared with industry partnership? Yeah, great question.
Yeah, Peter, thanks for that. It's venture capital, as you know, is a difficult source. The venture capitalists tend to want to take a lot of the company and take as little risk as possible. So they get a lot of asks and don't tend, in my experience at least, don't tend to give a lot of money. It's in the early stages, I would prefer to find either non-dilutive funding from grants or so on, which is also hard, or to go to family and friends who are going to be less greedy. Venture capital is a great bridge between the family and friends financing and the significant institutional raising that you'll need to do later as it goes. But it is expensive money in terms of how much capital you give up and how much control you give up. I'm happy for you to come back and give your response.
Thanks, Peter. And something that I'm wondering about here, that we've heard a bit about these challenges. I'm just wondering if you could say a little bit more about the problem solution validation challenges that are specific to synthetic biology and specific to this field that might not exist in other fields.
Yeah, the issue, of course, is taking a finding that's in the laboratory through to real life. So lots of things work in the lab, lots of things work in vitro, but being able to scale them up, being able to apply them to the situation in the clinic or in agricultural setting. There are lots of hurdles that are both man-made, i.e. things like the regulatory challenges around determining safety and efficacy, but also are biological. Things that work really well in the lab. For example, you can look at CRISPR-Cas9, or you can look at RNA interference, which works beautifully when you're looking at cells in the lab. The questions are, how do you tailor them and deliver them to specific tissues without getting off target effects and so forth? So those are challenges that revolve around biotech and synthetic biology that are less of challenges in mining or IT or other industries. So it's that scale up and taking from an exciting finding in the lab to the real world that present enormous complexities, which is why it's a very high risk space. Does that answer your question?
Yeah, it does, Peter. Thanks very much. Yeah. Jason, did you have a question or a comment?
Yeah, it's a bit of a roundabout one, maybe a silly one, but I just wanted to understand, like, thinking about what Peter said about the context of VCs and industry partnerships. And I just wanted to know, like, for a given kind of stage of a company, say, is there a difference in the kind of, I guess, milestones you might expect from a VC versus industry in terms of like what they would want to see from you at a given stage for them to be interested as customers? Does that make sense?
In my experience, and I'd be happy to hear Peter's view on this too, but in my experience, they're not hugely risk takers. So they like to have the technology, risk modified or risk lowered, which, as I said from my previous remarks to Jonathan, that can be complex and can be quite expensive. So in Australia, the venture capital market is less easy than it is in the US because there's fewer of them. simple solution. And in my view, it was easier to raise money in the US because the analysts in the US are usually subject matter experts, and therefore the fund managers bring in the analysts who are subject experts, and they drill you very, very closely on the technology. And if you can address that, then you go a long way to be able to raise the funds from the institutions. In Australia, most analysts tend to be generalists, and therefore the more esoteric the idea, the concept, the more difficult it is for them to be able to confidently advise their masters to invest. And the same, the venture capitalists tend to be a bit better in terms of their analysts, but they have a lot of opportunities, and therefore they tend to take on those that are less risky. But Peter, you got your hand up, so happy for you to comment on that.
Yeah, no, I agree completely, Peter. I mean, I think that it's in the nature of VCs in Australia. They tend to be looking for something very close toward the market. They're not interested in the long road to developing a product. They want something much closer to a product that they can then fine tune and take to market. Has been my experience. Yeah. And I think your comments about the US are right on the money, which leads me to ask you, do you think, in fact, in Australia, we should be, as major institutions such as universities should be looking at improving their pathways through to venture capital sources in Europe or the US, rather than focusing in what is really, it's a real small pond, it's a real small capacity in terms of fronting substantial funding in order to take ideas all the way through to the marketplace?
Sure. Look, in my experience, Peter, the US, I mean, I went to the US in 2013, having struggled to raise $1 million for the company that I was with a year earlier, to raise $5 million from there, with the idea of raising $5 million in Australia. 3 days later, having had around 25 meetings with institutional funds in New York, I had $52 million on the table, of which I couldn't take it all. We took 32 in the end, and I sat in the in a boardroom in the Chrysler building, signing a deal for $32 million after 3 days. Now that didn't happen really overnight. I had been to the US on numerous occasions talking about the company, but it all came together. That would never have happened in Australia. The US has a huge appetite for innovation and entrepreneurship that is lacking, sadly, in Australian funds in general. They tend to be very risk averse here, whereas in the US, yeah, they're happy to have a diverse portfolio of high risk and medium risk and low risk ideas or concepts. So I think you're absolutely right that having a real push to develop a network of those investors in the US would be something that the universities in Australia should definitely propagate. And I've got, as I'm sure you have, quite a list of potential investors that are interested in looking for new ideas.
Thanks to both Peters. Yeah, some interesting ideas there. Another question from Robin on the chat. In the earlier context of idea validation, what strategies do you propose for engaging with market incumbents, particularly when the market is dominated by small players like startups?
Yeah, so when you have an early stage idea, and the market is dominated by a larger player that may be threatened by that idea, it's an interesting challenge. I mean, you have to think about Kodak in this context, and how they ignored the digital era of photography, and in the end they got killed by it. So just because you're first to market doesn't mean that you're best to market. If you've got a significant technological advantage over the market incumbent, then you need to develop that to the point where you can prove that it works before you engage with the market incumbent. But by that stage, hopefully you've engaged with investors who can give you the oomph to be able to demonstrate that in fact it does work. And then the market incumbent has a choice. They can either acquire you, work with you, or ignore you. And then you have a choice.
Thanks, Peter. Emily, did you have a question or a comment?
Yeah, I would love to kind of maybe follow on, because I think, Peter, what you mentioned early on about, when you, in your early stages of your career, where you had ideas and you said, there's going to be lots of people who will say no or say, oh, that's not going to work. And particularly, I think that is often, I guess, one of the challenges for people who are working on, SynBio-based ideas. It's almost like it's impossible, right? You're sharing, you're almost creating visions of something that hasn't happened or never have happened yet. So I'm really keen to hear maybe some of your examples about, like, how do you differentiate between feedback from people who kind of just can't see the possibility versus hearing the feedback and going, okay, maybe I need to listen to that. And, what can I do to, sort of, is it these design experiments or get data that helps me, I guess, make sure that I am on the right track, rather than, sort of building something that you might spend 10 years building this wonderful product, but nobody really needs it or nobody really wants it. So yeah, maybe kind of share a little bit about how did you kind of approach that?
Yeah, Emily, look, that's a really interesting question for me, because I tend to be a bit pig-headed. So I remember distinctly when I had the idea to start Cryosite, which was to outsource ultra cold storage of biological material, which no one had done in Australia at that stage. And I talked to a sales rep from a company, and she said, "You've got to be kidding. That'll never work. No one will ever do that." A year later, she became the sales manager for the company, for Cryosite. I trusted my gut. I plowed ahead, started the company. Well, I started the company with a group of people. So we started the company. And once it was up and running, then she joined it. One way to do it is that if you're convinced that your idea will work, then ignore the naysayers and go and sell the idea to people who may listen to you, and prove them wrong. And ultimately, if it doesn't work, well, at least you've had a go. But gee, you don't want to die wondering. And whatever it takes to prove them wrong, or prove them, so whatever it takes, you can do small experiments, you can do large stuff, or you can just go and do it and see what happens. I mean, with Cryosite, we started the company, we went out and we talked to a whole lot of people, and eventually, slowly, it happened. And it now pays dividends. But gee, it wasn't easy. It's not easy. But if you're convinced that it's going to work, and you've done your research, with those questions that I showed at the start, and you're happy that it could work, go and do it. Peter, you've got another hand up.
Yeah, I was going to follow up on what Emily was saying. So especially in the area of a research technology that is, it's at an embryonic level. You see what the possibilities are, but the road, no one has really walked that road and put all the milestones in place. So here's my question, which I think is quite relevant to SynBio, and that is, do you need to create an academic buzz around an area to make it more attractive? So in other words, I mean, I get all you're saying about the importance of having patents in place, but at the same time you're doing that, to what extent do you think creating an academic buzz is important in making your product, your vision, attractive to potential investors?
Yeah, well, I think having some publications is an attractive asset for the invention, for a start. I mean, one thing that I do look for is to see whether it's been published and whether it's been, whether there's been follow-up on it and whether it makes sense. So, so I think that's important, but I don't know about the buzz, Peter. I don't know if you need it. I think it's, and I'm not sure. I mean, I think that in some cases, sure, it gets significant. If it's, well, think of cold fusion. That had a lot of academic buzz around it, and ultimately it didn't work. So so the proof of the pudding is still, does it work? The other thing is that there have been a couple of publications from industry that have shown that a lot of the academic publications couldn't be reproduced. Yeah. Which is really quite concerning from an academic point of view, and also from industry. So I think that whilst it's important to get the academics excited, it really ultimately it's the raw data that demonstrates that it actually is viable that is very key. But again, it's horses for courses. So in some cases, no doubt, an academic buzz will really excite the investors, and if it works well, then then you can get a very high bang for your buck. So
sorry, I'm just coming from the point of view of if you're looking at investors who've got knowledgeable people evaluating technology, to what extent does the existence of an academic buzz also facilitate the process, or is your argument that in the end, what they want to do is just look at what you've got? Does it look like it really has legs? Thank you very much. That's what really is that's the bottom line.
In my experience, it's more the latter, but certainly the academic buzz won't hurt. On the other hand, I was involved in a company that had publications, but could not be reproduced by any other groups. And so the lack of an academic buzz made it really difficult. So, it works both ways. Mm-hmm. So the academic community and involvement in it is certainly an advantage, but it's probably, well, it's an advantage for sure.
Mm. Let's call this to this part of the session to a halt now, and thank Peter very much for a really fascinating discussion here, drawing on huge amounts of experience. Thank you very, very much, Peter. What we'd like to do now is have a bit of a play with some of those concepts, maybe look at some tools and frameworks that might help us apply the principles that Peter's laid out, and kind of have a play with applying them to to ideas that that we might be kicking around ourselves. And this is how we'd like to spend the remaining time that we have together this evening. Just to get us started, so if we're thinking about tools, frameworks, kind of formal processes that we can use for validating the value proposition of the idea that we might be playing with, or the invention that we might have, who's come across the value proposition canvas as a tool? People come across the value proposition canvas? Yeah, okay. We're going to kind of look at some thinking that's kind of related to that. We're not going to dive directly into the value proposition canvas here, but that kind of thinking, being really clear on identifying what problem we're solving and for whom. Let's go into that in a little bit more detail. The idea that Peter's expressed pretty clearly is that we're looking for a situation where we have a clear understanding of what our eventual end customers need and care about, that there's an overlap between that and what we can do really well, and that not too much of that area is covered by existing competition. So we're really looking to understand how big that space is that's in the dark purple color there on the screen, that's got the arrow coming down to it from the top of the screen. We're not looking for the tiny little overlap area between the three circles in a conventional Venn diagram. We're looking for the overlap between what our customer needs and cares about, what we do really well, and the part of that that isn't already covered by our competition. So there's still an existing unsatisfied need that our customers have. And so here's a tool that we use for testing and validating our understanding of that space. So we're talking about designing experiments that we can do to check that there really is a fit between our solution and what the customer's looking for. And this is the concept of a minimum viable experiment. Because, of course, what we're always trying to figure out here in startup land is, if I need $800 million to prove this, then there's the chance that I'm going to waste the $800 million if I go off in the wrong direction. What's a way that I can do this really cheaply and really quickly, so that I can then go out and raise the money that I need to be successful? And here's a framework that we've used to do this, and this is one that fits in very nicely with this evening, given that we're talking about the 10x program, or SynBio10x in particular. We're looking for the areas where our idea has a significant advantage. Let's call it a 10x advantage. And so the framework that we'd like you to have a bit of a play with this evening is this one here. So we suggest, if you look at the box on the right-hand side here, what we're trying to do is articulate our customers, what they're trying to do, what problem they have in doing that, and what's valuable about our solution. And the framework that we're proposing that you use here is to state those things in this sentence. We believe our capability, our innovation, our solution, our invention helps these people, our customers, who want to do this job, what they're trying to do, by having these unique values. Here are the things that are unique about our offering, our capability, our solution, in helping these people do their job, and in particular, helping them address the barriers or problems that stop them from doing this job today. Let me give you an example of this. This is a team or a company, as it is now, called Progel, which spun out of the University of Queensland, and they work in the area of microgels or encapsulation. So this is the way that they described their assumptions about their 10x advantage. So when this company was first spinning out, this is the way they would have described themselves. We believe our industrial scale microgel encapsulation process will help probiotic beverage manufacturers, sales directors who want to increase consumer satisfaction and product sales by enabling probiotic count to be maintained until the end of product shelf life at comparable production cost to existing processes. So in this one sentence, admittedly it's a fairly lengthy one, Progel had identified their customers, the sales managers at probiotic beverage manufacturers. They'd identified the job that these people are trying to do, which is increase customer satisfaction and product sales. And implicit in this, they'd identified a problem that they believed that they had, which was to do with maintaining probiotic counts until the end of product shelf life. They believed that these manufacturers had difficulty doing this with existing solutions, and that their encapsulation would solve that problem. So that's the way they stated their assumptions about what they believed their value proposition was. Here's the experiment that they actually did to test it. And firstly, here's the concept. So what are we going to do to test the value, our value proposition? We want to do an experiment to test the fit between our solution and the customer's problem, to test that we've actually got this right. And we want to not just hear what the customers say, we want to watch what they actually do. So before we go into this, we define a kind of a pass-fail criteria. Our confidence in this fit will increase if we observe a particular behaviour. And we've really hit the jackpot if we can figure out a way to do this that we can do in a couple of weeks and will cost us a very small amount of money. So here's how Progel actually did this. They had some feasibility study results. They had that basic data that Peter was talking about before that showed that, or that they believed showed that their concept was feasible, that it could actually preserve probiotic counts until the end of typical end of product shelf life. So what they did is they sent these feasibility study results in summary to 10 sales directors of these target companies that they were after, and asked them to participate in a proof of concept co-design workshop with the lead researchers. And the experiment was simply, will people accept this invitation? Will they give up a day of their time to come and work with our researchers to actually design concepts that might solve this problem based on our research? They decided that they would be confident that they had a fit, that they'd actually understood the problem correctly, and that their solution was uniquely valuable in solving it if they got more than 50% of the 10 people that they invited who actually came to the workshop. This is clearly something that was doable in less than a couple of weeks and was not going to cost them very much money. So really nice example here of a kind of a gold standard minimum viable experiment. So what we'd like to suggest now, what we're going to do is break you into groups and put you in breakout rooms and give you the opportunity to play with this concept. What we'd like to do firstly is ask if you could identify yourself, maybe raise your digital hand on Zoom, if you're someone that actually has an idea, a SynBio idea here, an idea for a potential startup. If you're a potential startup founder, a potential participant in the SynBio10x program here, the reason that we want to identify those people is that we want to put one of those in each of the breakout rooms and have that person kind of lead the discussion here about, okay, what's my assumption about my value proposition, and what minimum viable experiment could I do to test that proposition? So if you're up for putting your idea on the table and having it kind of be tested by this process, please raise your digital hand as Jason has done. Is there anyone else here who's willing to take that plunge? Anyone else who wants to have a play with that tool applied to your idea or an idea that you're playing with? We appreciate that at this stage, some people might have an idea, but they might not quite be ready to talk with others about it yet. That's okay. We don't want to force you into a corner there.
Yeah, I don't really think I can participate in my own program, to be honest, even though the investment's so good.
So given that there isn't really anyone else that's willing to do that at the moment, we'll put Jason in a room with a few other people, but we'll separate the rest of you into a handful of small groups and just let you have a bit of a play with that idea. Maybe not necessarily applied to ideas that you're working on right now, but just want to give you 10 minutes or so to play around with that idea, this minimum viable experiment idea. Emily, anything to add to that?
Yeah, I think, you know, the whole idea of obviously you joining this session is you're interested around how do you do this? Maybe you do have an idea, maybe you know somebody who is working on an idea, and potentially following or maybe using a process or a framework like this might be helpful to them. And so when you jump into your breakout room, if you do have an idea, feel free to kind of dive in and share. But if you don't, maybe have a bit of a conversation with your new friend about how might this framework be helpful.
From our experience, and I guess it's pretty obvious, I mean, if you've got hundreds of millions of dollars, it's always easy to test these ideas. Just build the product and see if anyone buys it. The challenge here is in identifying what's the riskiest part of the idea, what's the riskiest part of that value proposition statement, and figuring out cheap and quick ways to reduce that risk as much as possible. That's where the real creativity comes in, and that's where some really clever thinking is sometimes needed. So we'd really invite you to be creative and kind of think broadly about some examples of how you might test those kinds of ideas. So we'll chuck you into these breakout rooms for between 5 and 10 minutes because we want to bring you back at the end to give us a chance to all wrap up before we send you on your way. So buckle up your seatbelts, and we'll see you back here in 6 or 7 minutes.